Nearly everyone knows that DNA fingerprinting can be used to prove a person's connection to a crime. DNA fingerprinting has become a very common term these days, helping to prove innocence or guilt in criminal cases and resolving immigration arguments and clarifying paternity. DNA fingerprinting is generally regarded as a reliable forensic tool when properly done. The techniques used in DNA fingerprinting also have applications in paleontology, archaeology, various other fields of biology, and medical diagnostics. Besides these, it is very helpful in biological classification as it can help to show evolutionary change and relationships on the molecular level. Moreover, it has the advantage of being able to be used even when only very small samples, such as tiny pieces of preserved tissues from extinct animals, are available.
DNA fingerprinting. Hello students, I welcome you all to today's lesson, which is about DNA fingerprinting. You all must be aware that every person has a unique fingerprint. An English geneticist, Alec Jeffreys, in the mid-1985s developed a technique called DNA fingerprinting. ਮੁੱਢਲੇ ਤੌਰ ਤੇ ਉਹ ਉਹਨਾਂ ਤਰੀਕਿਆਂ ਨੂੰ ਲੱਭ ਰਿਹਾ ਸੀ ਜਿਨ੍ਹਾਂ ਰਾਹੀਂ ਉਹ ਹੈਰੀਡਿਟਰੀ ਡਿਜ਼ੀਜ਼ਸ ਐਂਡ ਜੈਨੇਟਿਕ ਡਿਫੈਕਟਸ ਦੀ ਜਾਂਚ ਪੜਤਾਲ ਕਰ ਸਕੇ। ਹਾਊਐਵਰ ਲੋਕਲ ਪੁਲਿਸ ਨੇ ਉਸ ਨੂੰ ਇੱਕ ਰੇਪ ਕੇਸ ਵਿੱਚ ਮਦਦ ਕਰਨ ਲਈ ਆਖਿਆ ਅਤੇ ਉਸਨੇ ਸਫਲਤਾਪੂਰਵਕ ਰੇਪਿਸਟ ਨੂੰ ਉਸਦੇ DNA ਪ੍ਰੋਫਾਈਲ ਰਾਹੀਂ ਪਛਾਣ ਲਿਆ। ਕੁਝ ਹੀ ਸਾਲਾਂ ਵਿੱਚ DNA ਪ੍ਰੋਫਾਈਲ ਨੂੰ ਸਾਰੇ ਫੋਰੈਂਸਿਕ ਸਾਇੰਟਿਸਟ ਦੁਆਰਾ ਪੂਰੇ ਸੰਸਾਰ ਵਿੱਚ ਵਰਤਿਆ ਜਾਣ ਲੱਗ ਪਿਆ। DNA fingerprinting allows a criminal to be identified from any blood, semen, or even hair or dead skin they may leave at the scene of the crime. So आओ देखिए कि DNA fingerprinting है ਕੀ। What is DNA fingerprinting? DNA fingerprinting is the term applied to a range of techniques that are used to show similarities and dissimilarities between the DNA present in different individuals. This technique of DNA fingerprinting has become one of the most common ways of convicting criminals. Many people are now more informed about the process than they have been in the past, and there is an increasing interest in DNA evidence. DNA fingerprinting, which is also referred to as DNA profiling, includes one of several similar techniques that can analyze and compare DNA from separate sources. Today, we will learn many aspects about DNA fingerprinting or DNA profiling. But before this, let me share learning objectives for today's lesson.
Upon completion of this topic, learners will be able to define DNA fingerprinting, discuss importance of DNA fingerprinting, describe procedure of DNA fingerprinting, demonstrate various techniques used in DNA fingerprinting, discuss various applications of DNA fingerprinting, discuss the limitations and problems of DNA fingerprinting.
DNA fingerprinting is a test to identify and evaluate the genetic information of DNA of a person's cells। ਇਸ ਨੂੰ ਫਿੰਗਰਪ੍ਰਿੰਟ ਕਿਹਾ ਜਾਂਦਾ ਹੈ ਕਿਉਂਕਿ ਇਹ ਬਹੁਤ ਘੱਟ ਹੀ ਹੁੰਦਾ ਹੈ ਕਿ ਦੋ ਵਿਅਕਤੀਆਂ ਦੀ ਸੈਲੂਲਰ ਇਨਫੋਰਮੇਸ਼ਨ ਇੱਕੋ ਜਿਹੀ ਹੋਵੇ। ਇਸੇ ਤਰ੍ਹਾਂ ਇਹ ਵੀ ਬਹੁਤ ਘੱਟ ਹੀ ਹੁੰਦਾ ਹੈ ਕਿ ਦੋ ਵਿਅਕਤੀਆਂ ਦੇ ਫਿਜ਼ੀਕਲ ਫਿੰਗਰਪ੍ਰਿੰਟ ਇੱਕੋ ਜਿਹੇ ਹੋਣ। Like fingerprints, cellular information of a person cannot be changed by any known treatment. But the actual point of concern here is that on what principle this method is based on. This process is based on the fact that no two people, with the exception of identical twins, have exactly the same DNA sequence. Also, although only limited segments of a person's DNA are used in the procedure, those segments will be uniquely different and represent the complete cellular machinery of that person. The chance of two people having the same identical fingerprint is about 1 in 1 billion. There is an interesting fact regarding human DNA. 99% of human DNA is same in all individuals. It is 1% DNA that differs. This 1% DNA enables scientists to distinguish identity. Let me explain with the help of visual that why DNA fingerprint is so important. Let us see.
DNA fingerprinting is done to establish paternity. This test may be used to identify the parents of babies who are swapped at birth, solve crimes. Blood, semen, skin, or other tissue left at the scene of a crime can be analyzed to prove whether the suspect was or was not present at the crime scene. Identify a decomposed body. This is useful if the body is badly decomposed or if only body parts are available, such as following a natural disaster or battle.
ਜਿਵੇਂ ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਸਾਰਿਆਂ ਦੇ DNA ਦਾ ਕੈਮੀਕਲ ਸਟਰਕਚਰ ਇੱਕੋ ਜਿਹਾ ਹੀ ਹੁੰਦਾ ਹੈ। ਵਿਅਕਤੀਆਂ ਵਿਚਕਾਰ ਅੰਤਰ ਸਿਰਫ ਬੇਸ ਪੇਅਰਸ ਦੇ ਆਰਡਰ ਵਿੱਚ ਹੁੰਦਾ ਹੈ। There are so many millions of base pairs in each person's DNA that every person has a different sequence. Using these sequences, every person could be identified solely by the sequence of their base pairs. However, because there are so many millions of base pairs, the task would be very time consuming. Instead, scientists are able to use a shorter method because of repeating patterns in DNA. ਆਓ ਹੁਣ ਅਸੀਂ ਪਹਿਲਾਂ ਇਹ ਸਮਝ ਲਈਏ ਕਿ repeating pattern ਤੋਂ ਕੀ ਭਾਵ ਹੈ। VNTR Variable number tandem repeats are end-to-end repeats of different short DNA sequences which exist in multiple copies. Number of these repeats can vary from person to person. An example of a VNTR is GG80, 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 which is 4 tandem repeats of the sequence GG80. Every human being has some VNTRs or variable number tandem repeats. To determine if a person has a particular VNTR, a Southern blot is performed, and then this Southern blot is probed through a hybridization reaction with a radioactive version of the VNTR in question. The pattern which results from this process is what is often referred to as a DNA fingerprint. I know these all techniques will be understood only if explained through video, so let us see it.
Steps followed in DNA fingerprinting. There are various steps of DNA fingerprinting. These are as follows. isolation of DNA, digestion of DNA by restriction enzyme, separation of DNA by electrophoresis, denaturing of DNA, southern blotting. Southern blotting is designed to locate a particular sequence of DNA within a complex mixture. Hybridization with different probes. Autoradiography for detection. Let us check them one by one. Isolation of DNA. The first step is isolating the DNA in question from the rest of the cellular material in the nucleus. This can be done either chemically, by using a salt to wash the extra material from the DNA, or mechanically by applying a large amount of pressure in centrifugation machine. Digestion of DNA by restriction enzyme. Second step involves cutting the DNA into several pieces of different sizes. This is done using one or more restriction enzymes. Separation of DNA by electrophoresis. Next, sorting of the DNA pieces by size is done. The process by which the size separation, size fractionation is done is called gel electrophoresis. The DNA is poured into a gel, such as agarose, and an electrical charge is applied to the gel with the positive charge at the bottom and the negative charge at the top. Because DNA has a slightly negative charge, the pieces of DNA will be attracted towards the bottom of the gel. The smaller pieces, however, will be able to move more quickly and thus further towards the bottom than the larger pieces. The different sized pieces of DNA will therefore be separated by size, with the smaller pieces towards the bottom and the larger pieces towards the top. The next step is denaturing the DNA so that the entire DNA is converted into single strands. This can be done either by heating or chemically treating the DNA with highly alkaline solution.
Once the DNA has been denatured, a single-stranded radioactive probe can be used. The next step is blotting the DNA, which is done by southern blotting technique. Southern blotting was named after Edward M. Southern, who developed this procedure in 1970. Let us see how it is done.
In this technique, DNA molecules are transferred from an agarose gel onto a membrane. This is nitrocellulose or nylon membrane, which is pressed against gel sheet. Some weight may also be used to get better result. This setup is allowed to stand for 12 to 18 hours. The fractionated DNA present on gel sheet is transferred to a sheet of nylon. The next step is hybridization with certain probes. Probes are basically pieces of synthetic DNA that have been made radioactive and that bind to the size fractionated DNA present on southern blot. Usually, a single-stranded radioactive probe is used where denatured DNA present on southern blot matches with a sequence similar to that on the probe. The final DNA fingerprint is built by using several probes simultaneously. It resembles the barcodes used by grocery store scanners. Hybridized DNA are compared with labeled markers. Next step is autoradiography for detection. To preserve this data for long, an X-ray is taken of the southern blot after a radioactive probe has been allowed to bond with the denatured DNA. On X-ray film, only the areas where the radioactive probe binds will show up on the film. This is original nitrocellulose sheet and X-ray film of the same.
This X-ray sheet will allow various agencies to use for identifying a particular genetic pattern ਜੇ DNA ਆਈਡੈਂਟੀਫਿਕੇਸ਼ਨ ਨੂੰ ਸਿਆਣਪਤਾ ਨਾਲ ਵਰਤਿਆ ਜਾਵੇ ਤਾਂ ਇਹ ਬਹੁਤ ਲਾਭਦਾਇਕ ਹੋ ਸਕਦਾ ਹੈ। DNA ਪ੍ਰੋਫਾਈਲਿੰਗ ਤਕਨੀਕ ਦੀ ਵਰਤੋਂ ਐਗਰੀਕਲਚਰ, ਫੋਰੈਂਸਿਕ ਸਾਇੰਸ, ਪੇਰੈਂਟੇਜ ਟੈਸਟਿੰਗ, ਫੂਡ ਇੰਡਸਟਰੀ, ਮੈਡੀਕਲ ਡਾਇਗਨੋਸਿਸ ਆਦਿ ਵਿੱਚ ਕੀਤੀ ਜਾਂਦੀ ਹੈ। While human DNA fingerprinting has numerous uses in law and forensics, from verifying paternity to identifying murder suspects, this technique also applies to other organisms. Plants, animals, and even bacteria have unique DNA fingerprints. An increasing range of applications makes use of this fact. Let us study various uses of DNA profiling or DNA fingerprinting.
Application of DNA profiling or DNA fingerprinting. Setting up of genetic data bank. On realizing the potential of DNA profiling, genetic data bank had been set up throughout the world. Databases of the genetic fingerprints of criminals have been set up in many parts of the USA and Britain. To establish paternity, DNA fingerprinting technology helped in reuniting the lost children with their respective parents or vice versa who were separated during war, violence, or natural disasters. Solving disputed problem of parentage, identity of criminals, rapists, etc. In India, several cases of disputed parentage has been solved. In criminal investigations, the DNA fingerprint of a suspect's blood or other body material is compared to that of the evidence from the crime scene to see how closely they match. DNA fingerprinting has proved that Dhanu, the human bomb, was the real murderer of Shri Rajiv Gandhi, the former Prime Minister of India. Immigrant disputes. More or less in every country, there is the problem of immigration. Ultimately, the identity is not confirmed whether they have crossed the borders or are resident of that country. Therefore, the problem of immigrants can be solved through DNA data bank. Fighting diseases. DNA fingerprinting is being used to identify antibiotic-resistant strains. This helps doctors to select an antibiotic other than the one to which the bacteria are resistant, or consider a different type of treatment altogether.
These were the applications of DNA fingerprinting. Now I will tell you some examples where this technique has been really used. some interesting achievements of DNA fingerprinting. Identifying victims of 9/11 attacks. ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਸੀਂ 9/11 ਦਾ ਉਹ ਭਿਆਨਕ ਹਾਦਸਾ ਜਿਹੜਾ ਨਿਊਯਾਰਕ ਦੇ ਵਰਲਡ ਟ੍ਰੇਡ ਸੈਂਟਰ ਵਿੱਚ ਵਾਪਰਿਆ ਸੀ ਉਸ ਨੂੰ ਭੁੱਲੇ ਨਹੀਂ ਹੋਵੋਗੇ। ਹਾਦਸੇ ਦੇ ਬਾਅਦ ਉੱਥੋਂ ਕਈ ਦਿਨਾਂ ਤੱਕ ਦਬੀਆਂ, ਸੜੀਆਂ, ਗਲੀਆਂ ਅਤੇ ਕੁਚਲੀਆਂ ਹਾਲਤ ਵਿੱਚ ਲਾਸ਼ਾਂ ਮਿਲਦੀਆਂ ਰਹੀਆਂ। ਇਨ੍ਹਾਂ ਦੀ ਸ਼ਨਾਖਤ ਕਿਸੇ ਵੀ ਤਰ੍ਹਾਂ ਸੰਭਵ ਨਹੀਂ ਸੀ। ਇਸ ਹਾਲਾਤ ਵਿੱਚ ਦ ਨਿਊਯਾਰਕ ਮੈਡੀਕਲ ਐਗਜ਼ਾਮੀਨਰਜ਼ ਆਫਿਸ ਨੇ DNA ਫਿੰਗਰਪ੍ਰਿੰਟਿੰਗ ਟੈਕਨੀਕ ਦਾ ਇਸਤੇਮਾਲ ਕੀਤਾ। ਇਸ ਨਾਲ ਇਨਵੈਸਟੀਗੇਟਿੰਗ ਏਜੰਸੀਜ਼ ਨੂੰ ਬੁਰੀ ਤਰ੍ਹਾਂ ਸੜੀਆਂ ਹੋਈਆਂ ਬਾਡੀਜ਼ ਨੂੰ ਰੈਕਗਨਾਈਜ਼ ਕਰਨ ਵਿੱਚ ਮਦਦ ਮਿਲੀ। DNA ਸ਼ੋਹਾ ਪ੍ਰੋਜੈਕਟ। ਇਸ ਪ੍ਰੋਜੈਕਟ ਦਾ ਉਦੇਸ਼ ਉਜੜੇ, ਅਨਾਥ ਅਤੇ ਗਵਾਚੇ ਹੋਏ ਬੱਚਿਆਂ ਨੂੰ ਉਨ੍ਹਾਂ ਦੇ ਰਿਸ਼ਤੇਦਾਰਾਂ ਜਾਂ ਬਾਇਓਲੋਜੀਕਲ ਫੈਮਿਲੀਜ਼ ਨਾਲ ਮਿਲਾਉਣਾ ਸੀ। ਬੇਸਿਕਲੀ DNA ਸੋਹਾ ਪ੍ਰੋਜੈਕਟ ਉਨ੍ਹਾਂ ਲੋਕਾਂ ਦਾ ਜੈਨੇਟਿਕ ਡਾਟਾਬੇਸ ਹੈ ਜੋ ਹੋਲੋਕਾਸਟ ਦੇ ਦੌਰਾਨ ਆਪਣੀਆਂ ਫੈਮਿਲੀਜ਼ ਤੋਂ ਵੱਖ ਹੋ ਗਏ ਸਨ। DNA ਫਿੰਗਰਪ੍ਰਿੰਟਿੰਗ ਦੇ ਅੰਤਰਗਤ ਕਲੈਕਟਡ ਡਾਟਾਬੇਸ ਜੰਗ ਦੇ ਦੌਰਾਨ ਨਿਖੜੀਆਂ ਫੈਮਿਲੀਜ਼ ਨੂੰ ਫਿਰ ਮਿਲਣ ਵਿੱਚ ਬੜਾ ਸਹਾਇਕ ਸਿੱਧ ਹੋਵੇਗਾ। So the techniques used in DNA fingerprinting have wide applications. Besides these, this can be used in biological classification. It can help to show evolutionary change and relationships on the molecular level, and it has the advantage of being able to be used even when only very small samples such as tiny pieces of preserved tissue from extinct animals are available. But the real question here is that, is this technique beneficial or not? So let us study the benefits and limitations of DNA fingerprinting.
Benefits of DNA fingerprinting. Paternity test by DNA fingerprinting. The most important benefit of DNA fingerprinting is that there are strong similarities shown between genetic fingerprints of parents and children. This is a benefit because a child's genetic fingerprint is made up of half the father's genetic information and half of the mother's genetic information. This means that the bands of a child's genetic fingerprint will match the bands on both of their parents, making it possible to establish paternity and maternity tests. Here you can observe that how a paternity test is used to match a child with his biological father. When the child's DNA fingerprint was compared with his alleged father and biological father, it showed that child's DNA matches with the biological father's DNA. This means that the father on the right of the child is his biological father, while the one on the left is not.
Limitation of DNA fingerprinting. Along with the benefits, the technique has some limitations also. One of the main problems with the process of DNA fingerprinting is that the sample can be easily destroyed. The tiniest pieces of genetic junk can contaminate DNA samples, causing them to be useless. Although DNA fingerprinting requires a good sample to work with, this problem can be solved by using the newer technique called PCR. PCR, that is polymerase chain reaction, is a technique to amplify a single or few copies of a piece of DNA across several orders of magnitude, generating millions or more copies of a particular DNA sequence. PCR can use extremely small samples of DNA and produce a much faster result. Another limitation of fingerprinting is that the procedure is so complex and hard to read the DNA patterns that sometimes the juror finds the evidence almost invisible. Even then, DNA fingerprinting has immense applications in the field of forensic science, genetic biodiversity, and evolutionary biology. So students, this was all about our today's topic. I hope you all have enjoyed this topic of DNA fingerprinting. Let me summarize the entire topic for you. The technique of DNA fingerprinting was developed in England by the geneticist Alec Jeffreys in the mid-1985s. DNA fingerprinting or profiling is a technique that identifies the DNA from an individual without examining the entire 3 billion letters in the full human genome. There are various steps of DNA fingerprinting like isolation of DNA, digestion of DNA by restriction enzyme, separation of DNA by electrophoresis, denaturation of DNA performing a Southern blot, making a radioactive probe, creating a hybridization reaction, etc. DNA profiling technology has applications in many areas like agriculture, forensic science, parentage testing, food industry, medical diagnosis, etc. One of the main limitations with the process of DNA fingerprinting is that the sample can be easily destroyed. The tiniest pieces of genetic junk can contaminate DNA samples, causing them to be useless. Now I will check how much attentive you were in the class. All of you should try and answer the questions I am going to ask. And the first question is, define DNA fingerprinting. The answer is, DNA fingerprinting is the term applied to a range of techniques that are used to show similarities and dissimilarities between the DNA present in different individuals. The next question is, who developed the technique of DNA fingerprinting? The answer is, the technique of DNA fingerprinting was developed in England by the geneticist Alec Jeffreys in the mid-1985s. The next question is, why DNA fingerprinting is done? Answer is, DNA fingerprinting is done to establish paternity. This test may be used to identify the parents of babies who were swapped at birth. Solve crimes. Blood, semen, skin, or other tissue left at the scene of a crime by a criminal can be analyzed to identify the criminal among suspects. Here comes the next question. What is Southern blot? The answer is, the Southern blot is the way to analyze the genetic patterns which appear in a person's DNA. Next question. What is gel electrophoresis? Answer. The process by which the size separation, size fractionation of the DNA pieces is done is called gel electrophoresis. Next question. Complete the sentence. VNTRs stands for dash. And the answer is variable number tandem repeats. The next question is, list some applications of DNA fingerprinting. Answer. Setting up of genetic data bank. Solving criminal cases. solving disputed problem of parentage, identity of criminals, rapists, etc., and fighting diseases. Okay, that was about DNA fingerprinting. I hope you have understood the entire topic. See you in next class with a yet another interesting topic. Thanks and goodbye.